from the Siglab microscope. This is from the smart eye camera. The beauty of the device is that you use only the light source of the smartphone and the camera function of the smartphone. This is the unique point because you don't need external light sources so that you can bring it anywhere. You don't have to bring it, the batteries. You only have to charge the smartphone. Now, when you capture images, the data is only going to the cloud, so you can do the remote diagnosis, of course, and you can see the location. But uh, the you have to have a certain ophthalmologist background if you want to do the remote diagnosis. So that's what we're gonna do, building up the AI. That's what I'm talking about. So as you can see, there is a right now 24 scientific evidences, including one in India, collaborating with the Aravinda Hospital, which is showing that the ability of this device compared to the conventional microscopes, using the case reports using as a telemedicine and build up the AI. Now I'll show you about some about models. So we can start with the cataract. Of course, the cataract is a major cause of the blindness worldwide, and I will have a cataract in the future. Anyone have a cataract in the future? However, it is difficult to identify the cataract because of course, there's no diagnostic tool in the remote places, and the patient doesn't know that they will be cured with the surgery. Now, we collected the data in Japanese patient and performed the machine learning. And because the device is a smart eye camera recording the eye video of the patient eye, so that you can, record more data than the eyes. So, for example, if you take the 30 seconds of eye, you can get approximately 900 frames. So you don't have to, maximize the quality. You just have to record the more seconds. Now, the ophthalmologist, but however, you need the two models of this tool because if you record the patient anterior segment of the eye, no one knows that it is diagnosable or not. So, we have two stages of the machine learning model. Basically, the first model is the extraction of diagnostic frames, and then we grade the cataract. And after that, we use a grad cam to visualize. So this is a little bit bulky slides, but as you can see that sensitivity and specificity is quite high. Because of the high sensitivity, it is great for you to use in the screening. And for the grade diagnosis itself, it's of a quite high AUC, the area under the curve. And you can see that the GradCam, the AI is observing where we want to see, no matter what, if it's undilated pupil or dilated pupil. Now, in Japan, we use the different grading called the emulator grading. This is pretty much conventional grading. It's as similar as like a Log 3 grading, but it's held in 1967. And mostly this grading is used in not the ophthalmologists, but the primary care physicians. So same protocol, we gather as many videos annotated by the ophthalmologist, and then we perform the machine learning. Now, this is a quite high performance, interestingly, because you can see the high accuracy, sensitivity, specificity, and AUC. Same thing as the grading itself. As you can see that it can diagnose the mild and the severe cataract more easily than the others. Same thing as for the GradCam. No matter what, if it's myodartic or not, the AI is observing the cataract. Now, we can move on to the dry eye disease, which is a little bit difficult because you have to do the fluorescent staining. Of course, there is a non-invasive BUT that we will calculate, but now we are mimicking the conventional way. So we are using the fluorescent BUT to calculate it, and we annotate the breakup positive or not. And as you can see, this AUC is not that quite high because the we don't have a blue light field. I'm sorry, the filter to identify, and the diagnostic performance itself is not that high. But as you can see, that we did only record, we only recorded like about 80 cases. So now we're trying to expand the N. Corneal opacity. So this is a quite one of the blindness disease, but it looks like that the it's not that hard to identify the corneal opacity, but same principle, same procedure. And then at this time, we use the two things, that is to augment the data to extract the cornea itself. Second, we call the clahet to adjust the image contrast more easily to observe. Now the performance is quite high, as you can see that over 95% of all performance itself. And that we said we did the manual extraction, of course, with the no downsizing, and the clahe is a pretty good do for the pre-process. Now we are measuring the anterior chamber depth as well with the same principle using anterior segment OCT. We call the CASIA system. You can see that the MAE of the itself is quite good. So if the patient ACD is lower than 2.5, it could be the risk of an angle closure, but it can identify pretty good. Now, this is the last one for the corneal diameter. The same principle using CASIA to calculate white and white, and then the MA is about 0.2 millimeter. So we are trying to expand it to screening of the congenital glaucoma and ICL implantation. Now, so I think the time has come. So we're trying to find more, team to make much more AI for the screening purposes, trying to gather more patients to the ophthalmologist. So this is it for my talk. Thank you so much.
Thank you, Dr. Shimizu. I really liked your innovative technique of using a video recording to augment the information. So are you aware if anybody else is using this video technique or is it unique to your team?
Actually, this device is unique to my team because the device itself we developed. However, there's a tremendous numbers of the smartphone technology with the anterior segment, but interestingly, no one is using video. And I think the reason is that it is they don't have any idea to build two stage of AI. One is extracting diagnosable frame, and second is like cataract or whatever that you want to see. So these two combination will be the pretty good and pretty interesting point that we build.
I have one more question. So evidently there are different teams across the world who are working on this, and um, basically what will be the key point which would either say everybody use one particular app or everybody has their own app? What will, because unless we share it, share our basically when you develop something, either you'll publish it and then it will just be a paper, but other people won't have access to it. So how are we going with this? Because I feel just in this platform itself, we probably have at least 3 or 4 teams just today who are attending this, who are working on it independently. So where will we go with this in future?
Yes, so I think there's a 2 point. One is that we have to generalize it according to the different lamp photos or other devices. And that's what we are trying to do right now. And second thing is that we want to share the platform, not the device. So in the future, I believe that everyone has their own device, or or maybe everyone has their AI. But the tool is bulky and it just stick it into place. So if you can install this technique into the app itself and install it to for example, the slit lamp of the partial part of India, they can use this device as well. I'm sorry, they can use this technology as well. So that's what we are trying to aim.
Thank you. Congratulations for your work. It's very impressive.
Thank you so much, and we are keen to have many collaborators in India as well. So if there's anything, please let us know. There's Dr. Rohan.
We look forward to that. Yes.
As well, so please ask either of us.
Dr. Sridevi.
Thank you, ma'am. Thank you, Professor. And now I'd like to welcome Professor Tirumurthy Velpandian, sir, who is the head of our Department of Ocular Pharmacology at Dr. RP Center, AIIMS. Over to you, sir.
Thank you very much for giving this opportunity to be with all today. And indeed, the pleasure is mine today to appreciate everybody on the day of, I must congratulate on Women's Day. I'm very happy that, especially to all the team your organization, all majority of them are women organized. I'm so happy to see that here also. Well, today I'm just going to share with you a little bit about our developments, the innovations which happened in the last few years, especially drug delivery devices, as well as the understanding what we had towards eye. Earlier, that we thought the pharmacology is something, just putting in midriatics and myotics and putting some few eye drops, which are antibiotics, which have been made for some other diseases, have been just made it into eye drops and put it in the eye. But today we have a different type of understanding towards everything. And that is also giving us a good opportunity for the development. If you just look at the drugs, ophthalmic drugs market in 2024 is growing rapidly, and strong growth in ophthalmic drug market, which is a very good thing for us to go for innovation, trying to bring new therapeutics. It's almost a compound annual growth of 8.3% in which it is going on, and I think now the world is looking forward for innovation in eye, especially ocular therapeutics. So we feel very happy about it. The first one which came up in our mind about when we just look into the drug trial of Allergan, especially about the Mementin. The Mementin is an excellent drug when it's been approved for many of the neurodegenerative diseases, especially Parkinsonism, etc. So the excitatory amino acid pathway has been very much implicated in neurodegenerative diseases, in which glaucoma is something which is falling inside our interest. So when the paper with this has been done, the moment it has been approved for Alzheimer's, and quickly the companies jumped into it, go for phase 3 clinical trials, especially in case of glaucoma. After giving so much of money, about $115 million, finally the music of the day is dead. So, what happened is that one of the editor of drugs R&D asked me to completely review it to know what went wrong, as per my view. So by the time, we had a very good knowledge about understanding about eye, how do we deal with the eye? It's much more specific. It behaves like an island. It's more independent. It allows only things which is specific. So we came to know about the involvement of drug transporters across the blood-ocular barriers. Then we understood the failure of Mementin in this case. So now we understand, by and large, unless you study the pharmacokinetics of a drug for eye, it's not easy for anybody to optimize. So all our efforts went on understanding which type of drugs can go in. When you give systemically administered drugs, and if you administer drug intravitreally, which type of drug can stay in and which will be pushed out. So now we have a very fair understanding about it, and I'm very happy that we took a leading role in coming up with the understanding towards this. Now, see, this is one of our classical publication which came in 2009, which shows that a drug which is administered inside the vitreous and along with the blocker of the pump, this gamma imaging shows that how it is getting distributed. You can see the kidneys are just brightly shown here. Whereas when we inject the same thing along with the blocker of the pump, it stayed for the long duration inside the eye. That was the biggest achievement what we had. We could document it visually. So we little bit concentrated on how to handle the blood-ocular barrier. Sometimes the drugs are more specific, sometimes they are transporter specific. So we started working on drug delivery systems. And that we fairly now see there are several drug delivery systems in the market, right from vitreous ganciclovir to Asudex, that is dexamethasone, several drug elution things, Ivation, etc., which came into picture. So to recently that RPC innovation which took place, one of our bright young faculty member, that time he was friend, and I must appreciate him. He has become a full professor today. So he came with an idea of why shouldn't we create a port for repeated administration of intravitreal injection, where that's a reason that many of the endophthalmitis comes up in case of diabetic retinopathy as well as AMD. Therefore, we created this small invasive implant is an intraocular surgery. In the sclera, it is implanted as a port. It's covered with conjunctiva. And many times this is a rabbit image showing that repeatedly we administered for 3 months. And it was very nice. The rabbit was not getting infected. That has given it's been recently, patented by RP Center. And this is an excellent system which came in our innovation. I hope with the clinical trials, if it proved to be good, we have an excellent device for repeated drug administration. There's another innovation which came up here. Professor Titial was involved with this innovation. What we did is that we made a small drug-eluting matrix, that is natomatrix, which elutes natamycin over the period of time, which is implanted intrastromally. It's again it's a IP is ours. This is something a revolution. One, I think you must have heard about it so much. Professor Namrata Sharma has done a clinical trials. We have done RCT, and we have given injected the natamycin, as you know very well, that it is insoluble drug. We injected intrastromally. This is again another one of our innovation. Protected by us by using site inclusion complexes. Exactly. So this is Zol and Natasol. We could see that they are capable of increasing drug penetration across the cornea very well. We could make a water soluble natamycin, and we could study that it is having very good penetration. So next one important one is that about extemporaneously prepared drug solutions, especially the drugs we compounded for specific purposes. This has been going on everywhere, every clinic, wherever we go that we have been asked, but there's no solution because these drugs are pharmaceutically unstable. Even for drugs like liposome, nanoparticles, they cannot be used because they're unstable in nature. The moment you put it in water, they start releasing the drug. So to solve this problem, we came up with another very innovative device called TransDecon, and through this device, we made all things possible. Today, I think now, whatever the privilege we have in RP Center, we are proud to share this with the whole India and the whole, on the African countries, wherever this kind of problems are very high. This guy's recently got a US patent also. And, we tested in the field testing. Somebody who doesn't even know how to make eye drops and things like that, he could make this drug preparation very simply, and they can put it. And that's the way that we wanted the drugs or first line defenses or unstable antibiotics or drug resistant cases we can bring into picture. And this is to summarize that ocular pharmacokinetics is a governing factor complicated by pharmaceutical stability issues. And drug delivery systems, fairly we know there are contact lenses, polymer shields, drug soaked, intraocular lenses, etc. The delivery devices, apart from delivery devices, we have got sub-arbitrary port for repeated vitreous administration, TransDecon for drug administration constitution. These are some of our recent innovations which came from RP Center. Thank you very much for giving this opportunity to share with you all about a recent development. I thank all my collaborators as well as my students for making this possible. Thank you very much, madam, for giving this opportunity. Thank you.
Thank you so much, Dr. Pandyan. That was a wonderful lecture.
So kind of you, ma'am.
Yes.
Thank you, ma'am. Thank you, sir. I now request Professor Tadao Maeda to come over. Sir is a distinguished research development manager with over 20 years of experience in the biotechnology industry and academia, specializing in retinal regenerative medicine. With extensive expertise in ophthalmology and molecular biology, he has made significant contributions to the development and clinical application of iPSC-based therapies for retinal degeneration, including ARMD and retinitis pigmentosa. Throughout his career, Sir has been committed to addressing unmet medical needs through innovative research and leadership in clinical translation. He plays a key role in international collaborations, bridging academia, industry, and regulatory bodies to drive advancements in regenerative medicine and ophthalmology. Over to you, sir.
Thank you very much. Can you hear me?
Yes, sir.
Okay, great.
Yes.
Okay, so thank you very much for your generous introduction. I would like to also express my gratitude to Dr. Tandon, Dr. Sharma, and the AIIMS committee members for this invaluable opportunity. And congratulations on the 58th Foundation Day celebration. Today, I will be speaking about the advance and the future prospect in iPS cell-derived retinal cell therapy for retinal degeneration. So first of all, I would like to introduce background information of our research. So we are focusing on the outer layer of the retinal pigment epithelium and photoreceptor cells. Those two cells play a fundamental role in our vision. Our retinal degeneration involving retinal RP and photoreceptor cells is nowadays one of the leading causes of vision loss in the Western countries, Japan, and could be in countries in Asia as well. So here is a color fantasy image of representative outer retinal degeneration, retinitis pigmentosa, and age-related macular degenerations. Retinitis pigmentosa starts from the peripheral region of the retina, and the degeneration proceeds to the center of the retina eventually. There are two types of AMD, wet type and dry type. Wet type is characterized with a colloidal neovascularizations here, and the dry type is characterized with RP and colloid degeneration, followed with photoreceptor degeneration. The bottom images are a visual field of late stage of those diseases. So retinitis pigmentosa, so central vision with small size of visual field, and AMD peripheral vision is maintained. However, central vision is severely disrupted. Both of them are devastating and have huge negative impact to the patient's QOL. So here is RP manufacturing method from iPS cell to RP cells, which takes 34 days. The strength of this method is the purification of the harvesting only qualified RP cells colonies. So due to the robustness of this method, highly purified RP cell can be obtained from the different iPS cell lines. So far, we have tried more than 20 iPS cell lines. We obtained the same quality of RP cells. The RP cells are stored under frozen condition until formulation as a RP cell strip. As quality assurance of RP product, we evaluated characteristics of iPS RP cells with various methods. The purity is the most important target profile of our product. The purity is more than 97% every time. So far, we tested 3 types of formulation: RP cell sheet, cell suspension, and RP cell strip in clinical research. Then we concluded RP cell strip is the best choice because this formulation have advantage of RP cell sheet and RP cell strip, RP cell suspension. So this movie is a show mode of action of RP cell strip. So RP cells gathered at strips under the group of a cell culture plate, and RP cell strip is loaded in the cannula, then transplanted subretinal space. The transplanted RP cell strip is spread out and forming a monolayers, like RP sheet. Then retina is revived after RP cell transplantation. So here is a representative clinical result after allogeneic iPS cell derived RP cell strip transplantation. The first case was 59-year-old female with dry type AMD. From 6 months after RP cell transplantation, retinal sensitivity in the edge of a transplanted region and the improvement of QOL was shown. To change the subject, this is the MOA movie presenting retinal organoid transplantation for retinitis pigmentosa. So retinitis pigmentosa are losing the photoreceptor cells gradually. We transplanted retinal organoid subretinal space. Then retinal organoids are matured under subretinal space after transplantation, then making a synapse connection with the host secondary neural cells. Before transplantation, there is no light sensitivity in the retina. However, after transplantation, photoreceptor is recovered, then retina is improved retina sensitivity. Then so far, we transplanted 2 retinitis pigmentosa patients, such a kind of retinal organoid, then partial recovery of visual functions obtained. So potential issues in the implementation of regenerative medicine could be solved by AI in the hospital model. So we utilize an AI-driven systems to evaluate RP cells adhesion region evaluation after transplantation in the patient eye as a primary endpoint in our clinical research. So this model is this system is developed by a deep learning system. And one of the challenges of the cell therapy is manufacturing consistency. So recently, we successfully moved our work to the humanoid robot called Mahoro. So with the application of this robot, now we can make the cells 24/7 and have easy tech transfer to the anywhere. And also to share the technology and the system generally, so we developed the hospital system. So we have already started collaboration with some countries in Asia and the USA. And also, so we established a registration system to evaluate clinical cases to select the proper cases for regenerative medicine. So this is the last slide of my presentation. Thank you for your attention. I would be happy to take any questions. Thank you.
Thank you so much, Dr. Maida. It was a very fascinating presentation to have this much level of success in situ proliferation of these RPE cells. And when you mentioned the iPSC cell derived RPE cells, would you say that your group is unique to have reached this milestone?
Could you say that again?
The iPSC derived retinal pigment epithelial cells to this level of sophistication, is your group one of the few people who have been successful, or the only people who have been successful for this ability to produce RPE cells from iPSC cells?
Yes, and iPS cells, there are, as far as we know, so just 2 groups succeeded. And in our group plus Dr. Kapil Bhatti's group in the NEI.
And
so other groups are used are not iPS cell, but they are used ES cell.
Right. And the injections were in animals or in humans?
So we already tried in the clinical studies. So we transferred it in the human with age-related macular degeneration and retinitis pigmentosa.
Excellent.
Thank you.
Ananya, would you like to invite the next speaker, please? Is Dr. Sujata here? So we're waiting for Dr. Sujata. Meanwhile, we can take a few questions if anybody has.
Ma'am, I'm just sharing my screen. Yes.
So Dr. Sujata is here. Ananya, will you please introduce?
Can you speak? Is the one?
So it's a pleasure to introduce Professor Sujata Mohanty. She is in charge of the stem cell facility at AIIMS. It is a center of excellence for stem cell culture and research. So the talk is going to be emerging therapeutic strategies and translational advances using stem cells for ocular diseases.
Ma'am, just sharing the screen. I'm sorry about that.
Yes, that's fine. That's fine.
Very good afternoon to everyone. And thank you, ma'am, for giving me this opportunity. Am my screen visible? Full screen?
Yes, it's not in the slideshow mode, but it's visible.
But now it's on full screen. Yes.
Thank you so much. Is it there? Thank you so much. Yeah. So good afternoon, and this is a specialized part of the AIIMS where we do stem cell research along with my all collaborators. And if we see the clinicaltrial.gov that is showing us the current scenario, there are approximately more than 1,000 clinical trials which are ongoing in the area of stem cells and ocular disease. So keeping that in mind, so we had a very fantastic collaboration with RP Center AIIMS in both clinical research, basic research, and preclinical research. So I will just show you some of the snapshot in different areas of ocular defect and diseases which we have embarked upon with collaboration. Just to say that all the clinical trials were held in processing of the cells was done in GMP clean room areas. The first one we initiated was corneal epithelial defect. And these are the multiple publication along with Professor Radhika and Professor Namrata. And the students involved was Shweta and Sudeep. So initially we did our processing of limbal samples using amniotic membrane as a scaffold. And we standardized the processing and the storage of these human amniotic membrane. And we found that they are quite stable at -80 for 1 year. So after that, we did some of the bench work in terms of their molecular characterization and quality control and quality assessment. After clearing that, we embarked upon the clinical trials. So in clinical applications, so this is the picture which shows that the limbal biopsy sample which reaches to the lab, and we grow them lab for 14 days. And with QC/QA, it is given back to the surgeon and it is transplanted back into the patient. And these are the few snapshots of the samples. This is the donor eye without any major effect on taking the biopsies. And this is some of the outcomes. And we have done more than 3,000 cases. So in terms of where both the eyes are affected, just an example of Stevens-Johnson syndrome, we try to harvest mucosal epithelial cells and were very much interested to see how does it cover the corneal opacity. So here, along with Dr. Radhika, Dr. Sudeep was the main scientist, and we took biopsy samples from the oral mucosa with the help of ENT surgeons, and this tissue was grown in the lab and was also done extensive characterization in terms of oral mucosal stem cells. So after that, we performed around 30 to 50 cases, and these are the few pictures of before and after 1 month. And the second one is after 20 months that the ocular surface has improved. So likewise, from the cornea, we also did some cases of age-related macular degeneration and retinitis pigmentosa. This was done in collaboration with Dr. Atul. Basically, the initial result was saying that there was no further decline in the visual acuity during 5 months of follow-up. And also in the retinitis pigmentosa, we found there was an increase in the improved central retinal function. From there, we was also very much interested to see that what are the other areas we can take on in terms of cornea. And the endothelial defect was the other area which we tried to focus on. And there was an initial collaboration with Professor Anthony Aldev, where we tried to harvest human corneal endothelial cells as an explant and also as in suspension culture. We did their molecular characterization based on their markers, ZO1 and also connexin 43, which are the gap junction protein. And we found that these endothelial cells, they maintain their phenotype. And now with Professor Namrata Sharma and Suman from my lab, we are trying to expand these human corneal now endothelial cells. And we have got some success, and we would like to first test them in an animal model before going for the clinical trial. We also did some innovation in our lab. First is, can we have an artificial cornea? So we had a PCL membrane, which we had done the electrospinning, and we compared it with the human amniotic membrane. We found that major problem was its opacity. Then we went ahead and did some plasma coating of this membrane, and it became translucent. And this membrane was further taken up by our MD student Raghav, and he did some animal work, which in the rabbit model of alkali burn, the outcome was fantastic, that it's biocompatible, it has good transparency and mechanical strength, and also shows no immune reaction. So we are thinking of taking into the first-in-man study. Second was like modifying the human amniotic membrane. You can see it has shown a clumpiness. So we used an innovation in terms of electrospinning, which is said here, like spraying some biomaterial over this human amniotic membrane. And now we get a membrane which is very much easy to handle. This is the picture of the novel membrane which we have transformed. You can see here it is very easy to pick up, no tearing, and it is easy to spread over the eye and the skin. The second one is like we are trying to have multiple form of this amniotic membrane, like sheet, solution, powder, and sponges, so that we can have various form of this amniotic membrane for the healing of the eye as well as for the skin. Second is in collaboration with IIT Delhi, Dr. Saurabh Ghosh and Dr. Radhika. So can we optimize the use of cornea in terms of decellularizing them and doing the cross-linking with chondroitin sulfase so that we can use them? So we started with goat cornea, and we did decellularization and the cross-linking with chondroitin to maintain its ECM. We did some in vivo validation in animal model, and we found that it is perfectly compatible and very good for the growth of the cells. So we may take this forward also in first-in-man. As I said that my lab is working on stem cells. So these stem cells also secrete some small particles which are called extracellular vesicle, and they are the powerhouse of in terms of cytokines growth factors, and these are very small, like 30 to 200 nanometer. And one of my students has opened up a company and selling these extracellular vesicles derived from the stem cells. So now in our lab, we are trying to have a bioink which can encapsulate these extracellular vesicles, maybe as a artificial cornea which has got stem cell properties from these extracellular vesicles and may be used further. So the way forward is like we have induced pluripotent stem cells in our lab. We may go ahead and differentiate them into particular corneal or endothelial lineages. We may integrate stem cells, extracellular vesicles in tissue engineering to make artificial cornea, and also to optimize the donor cornea, which are not suitable for clinical purposes. So in the end, I would like to thank all my students and also the RP Center, especially Dr. Professor Radhika Tandon, Professor Namrata, and Dr. Atul, who were a wonderful collaborator, and our collaborator from IIT Delhi, Dr. Ashmi Agarwal and Manjeet Jasal. So thank you so much.
Thank you so much, Sujata. That was a very wonderful overview of all the expanse of work which is going on at the stem cell facility at AIIMS. And it has been a path-breaking facility to provide this service across research as well as clinical applications. And not only at AIIMS, but also some of the other institutions who don't have these facilities, they can also avail of these opportunities. And if I can ask that, sometimes there has been a lot of variation in concept and policy regarding what is going to be the regulation. So we have certain things were taken as drugs, then we have stem cells. So in between, some of this cannot really be, certain aspects of it, like for example, the corneal decellularization work or even the limbal cell work, it can also be cell and regenerative therapies. So which direction are we going for, say, one can say registering such procedures or products or recognizing them as standard therapies, or say Sadisco approval, whichever agency is involved in qualifying that a particular product or procedure is okay for patients, because at the present we're all working in clinical trial mode or study mode or research mode. But eventually, where will we go with this?
Ma'am, very good question. So recently, as you know, we faced a lot of problem in making them applicable and available to many patients apart from the research mode. But now CDESCO, which is a part of DCGI and ICMR, they have come up with renovation in their guidelines. So there will be academic trial, and also there will be a way out to have early translation of these findings, which is done by the academic institutes. And second is like they are going to help us in pairing up with some of the commercial companies to take whatever the benchwork we have done to the bedside. So they have opened up in those scenario to fast track whatever finding which we have done in the academic institute and take it to the commercial level as well as to the patient. There are few changes which is going to happen soon.
Okay, that's good. So we have Kaniz here from MEJ, and we can ask that what is the policy being followed in Japan? Do they have any government regulations for the cell and regenerative therapies? Dr. Kaniz, or because we're going to have a discussion on this in the brainstorming. Otherwise, we can go ahead with our next talk. We have Dr. Nishat Hussain, who is the head of microbiology at RP Center. And Dr. Nishat is going to cover multiplex PCR for detection of herpes simplex virus. Over to you, Dr. Nishat, please.
Thank you, ma'am. A very good afternoon to all present. I'll be speaking about a multiplex PCR, which we developed in our section for the detection of herpes simplex viruses type 1 and type 2, cytomegalovirus, varicella zoster virus, and adenovirus in ocular viral infections. I declare no financial interests. So about viruses, we know that they can infect any part of the eye, and they can range from simple self-limiting discomfort to possibly vision-challenging manifestations. And not only for the management of the presenting complaint, we need to know the accurate diagnosis for specific sequelae and complications as well. So the most common viral pathogens in eyes are herpes simplex viruses type 1 and 2, cytomegalovirus, varicella zoster virus, few serotypes of adenoviruses, and enteroviruses. We also know that conventional methods are not of much help because of their various limitations in detecting viruses, and polymerase chain reaction has proven to be a valuable technique and has become quite popular recently. Now, in polymerase chain reaction, what we do is that a signature genetic component, a signature genetic target of each pathogen is identified, and then it is amplified with the help of these steps of denaturation, annealing, and elongation. And when multiple copies of the signature genetic component have been created, then with the help of post-PCR techniques, for example, gel electrophoresis, we can detect whether the specific target was present or not. So thus a diagnosis is made. However, PCR has a limitation that for each pathogen, we have to put a different reaction. And this becomes when in the differential, there are a lot of pathogens, it becomes very time consuming, expensive, cumbersome, and error prone. So these limitations to a certain extent are taken care of by multiplex PCR, in which 3 to 5 targets can be diagnosed in a single PCR, thus saving on time, cost, and hands-on work. So if we have 3 pathogens, for example, in differential, for which we are going to put 3 reactions to obtain results like this, it is possible with the help of multiplexing that in a single tube, all the consumables, target primers, and reaction conditions are devised in such a manner that we are able to obtain results like this, all the targets in a single reaction. So with this background, we move to our objectives that we wanted to establish a multiplex PCR for detecting ocular viral infections. However, we could only multiplex DNA viruses, including herpes simplex virus 1 and 2, VZV, CMV, and adenovirus. Enteroviruses being RNA viruses could not be multiplexed. So we first standardized 10 uniplex PCRs, 2 for each of the viruses of interest, and then these standardized PCRs were checked in control strains and known positive specimens for different study viruses. These were the controls which we used. Next, we moved on to multiplexing. So in multiplexing, the uniplex PCRs were run at different annealing temperatures and with variations of cycle conditions, as well as various multiplexing combinations of the PCRs were put. We wanted to get the best reaction conditions and the best targets for all the viruses of interest. And that we were able to obtain. We got 5 targets, annealing temperature, and reaction conditions, giving best results, and they were finalized for multiplex PCR. These are the primers of the 5 targets which were finalized, and these were the reaction conditions which we used for the multiplex PCR. And this is the multiplex PCR in which all the 5 viruses which we're looking for could be obtained in a single reaction. Next, the multiplex PCR was again run with control strains and known positive clinical specimens for different study viruses. And then we did uniplex and multiplex PCRs for the study samples and controls. Initially, we did 170 samples and we got very good sensitivity and specificity. Not only that, all the 5 viruses could be identified directly from the eye specimens. Also, we got very good results with non-invasive samples like conjunctival swab and tear samples. Also, we got very results with posterior segment samples like uveitis and chorioretinitis cases. The multiplex PCR was immediately put to use for diagnosing ophthalmic viral infections in our patients, and these are our real-time results that in case of corneal scraping or tissue, herpes simplex virus is the most common virus identified, followed by adenoviruses, and in case of conjunctival or corneal swabs, adenovirus is the most common virus, followed by herpes simplex viruses. So when faced with the bigger challenge, because of the wherewithal in place, we could do the microbiological workup for most of our cases. And these are the key results of the outbreak that adenovirus strain 8 and enterovirus, which was Coxsackie virus A24, were the major driving pathogens in the outbreak. And also because we were doing the multiplex PCR for all our samples, we could detect herpes simplex viruses also in 3% of the cases. Thank you for your patient hearing.
Thank you so much, ma'am. I now request our chair, Professor Radhika Tandon, ma'am, to give her inputs.
Yes. Thank you so much, Nishat. That was a wonderful talk, and many congratulations on this excellent work that you have been doing with your team.
Thank you so
I had
much.
wanted that there will be a lot of variations in how these facilities are used in different parts of India, also and in different countries. So, what is the current status in the world for using these techniques? Is it going into routine, taken as a routine test for routine clinical work, or is it more in a more high research mode?
Actually, ma'am, for viruses, whatever institutes or laboratories are detecting viruses in ocular infections, they are using PCRs only. But the thing is, they are using single PCRs for each of the viruses of interest. So we have gone a step ahead by multiplexing majority of the viruses. Enteroviruses, which are again a very common pathogen in eyes, we have not been able to multiplex because this is an RNA virus. However, in 2 reactions, one for all the DNA viruses and one for RNA viruses, we are able to detect almost all the pathogens.
So that's remarkable and actually quite a breakthrough. And maybe you can consider making it available as a kind of we can try to see if there is some platform, commercial platform, which will be willing to make this available in the market. So we can consider that because this is a very big breakthrough.
Yeah, that is a very good idea, ma'am. If you are able to do that and make it available for others also who have the facility but not the multiplexing. So that could be done. Thank you
much for giving MEJ this opportunity and an opportunity to be part much for giving MEJ this opportunity and an opportunity to be part of this great event. And we are really pleased to be part of this RP Center Foundation Day. And from our side, the 2 experts attended in this session, and they also shared some of their activities, what they are doing and how Japan is doing to some extent. And at the same, actually from Japan side also, the Regenerative Medicine Act is now the government and it's a national policy to promote the advancing regenerative medicine as well from Japan side. And during our last visit in January, our CEO emphasized that from MEJ, we had initiated the ME India initiative to, foster the collaboration in the health sector. So we are very much pleased to have this platform with AIIMS, and we really hope that in the upcoming brainstorming session as well, we will come up something to collaborate together with Japan, so that we both the health sector in India and also in Japan can take the opportunity to advance using the advanced innovative solutions from Japan. And we hope that the solutions Dr. Maeda and Dr. Shimizu shared, and through the brainstorming session, we can come up. And there are other regenerative or cell therapy in ophthalmology as well. We could come up and make it a tangible partnership, considering, healthier India. Thank you.
Excellent, very encouraging. Thank you everybody for participating. We are now shifting to the auditorium because we have the memorial lectures in the names of all our previous chiefs. So we look forward to meeting again on Monday in the brainstorming. Okay.
Yes,
Thank you so much.
I will
Thank
be
you, everyone.
collaborating with the team. Thank you so much.
Thank you.
Thank you again.
Bye-bye.
Thank you, Dr.
very much.
Shimizu and
Thank you,
Dr.
everyone.
Maida.
Thank you.
Thank you. Thank you.
Bye.
Thank you, Sensei. Thank you so much.
Recording stopped.